Good morning, everyone. Thanks for the introduction, Sophie, and the invitation. It's my first time in your space, so I'm really happy to be here. Today, what I wanted to talk to you about is a subject written up there, which is how breathing impacts neural functioning in the mouse, and how this in turn can help the animal regulate its fearful behavior. But this topic is sort of a zoom in on a particular topic within a much broader field, which is how our body can feed back to the brain and impact our emotions. And so I wanted to start by introducing the subject in this context, particularly because a lot of what we know about this, both theoretically and experimentally, has fed from human theoretical work into the animal work. So I'll start with that. In a way, it's quite lucky in this field because it's very easy to sort of plant a chronological flag in the timeline of where the debate about the role of the body in emotion started. And that is in 1884 with a very famous article written by William James, and that was swiftly backed up by another researcher. And James posited a very strong relationship between the body and emotions. And I've put a quote of his paper there, which I'll read for you. So what James proposes is that bodily changes follow directly from the perception of what he calls the exciting fact, the emotional stimulus, and that our feeling is the same change as these same changes as they occur is the emotion. So he says that the common sense view, as you see over here, is that we see a bear, we're frightened, so we run away. But he instead posits that we are afraid because we tremble or because we run away, as illustrated here. So two things to take from this quote. The one is that he's positing really a one-to-one relationship between what's going on in our body and our subjective experience of our emotions. And the second is that it's really a hypothesis about the causal chain of events. Is there an emotional stimulus that evokes an appraisal, for example, of the threatening situation and evoke both the subjective state and bodily somatic reactions? Or instead, does the emotional state follow from the somatic and bodily reactions? And I think one of the main appeals of his proposal is that it makes a lot of introspective, intuitive sense, because we've all had very strong experiences, I think, how tightly associated our bodily state is with our emotions. If we're a little bit stressed, we can hear our heart pounding. If we're a little bit bothered or ashamed by a situation, we'll feel our cheeks glowing red. However, of course, these ideas have evolved a lot and have been placed in a broader context that questions the directness of this link between the body and the emotional states. The first point, of course, is that James was almost exclusively focused on the subjective experience of emotions, which, particularly for animal research, sort of alienated his hypothesis to being investigated in animals. And of course, it's really crucial to point out that we don't just experience our emotions. Our emotional states influence our behaviors, the way we make decisions, the way we perceive things. They're going to propel us to adapt our behavior at different timescales, our memory at different timescales. And these, of course, thankfully, are things that can be readily measured in a more objective way in humans, but also in animal models. So we're not just going to be interested in subjective emotions, but more broadly in emotional behaviors. And there's another change that's intervened that I think is very important. That's the fact that James posited this very direct link. But of course, we have a dedicated system in the brain to sensing the state of our body, interoception. So it's the sense dedicated to sensing our visceral body, our heart, our breathing, our blood pressure, states of our digestive system. And many people have pointed out that for this to be transferred to an emotional subjective state or to an emotional appraisal of behavior, it's very likely integrated with other things, what we know about the context, our beliefs about the situation we're in. And I've just sort of highlighted two theoretical frameworks for thinking about this. They're quite different, but they both have the virtue of underlining the fact that the sense of our body has to be integrated with much more information in order for the emotional state to be inferred and to act upon our behavior. So this is a slightly more complicated picture that I think reflects a little bit more how people think about these relations since James' initial proposal. But of course, criticisms have been aimed at this model, and they were aimed at the model really initially by Walter as a researcher also working on emotions, but not in humans. He was focusing on cats. and he was working on the sympathetic system. And very soon after James's article came out, Cannon wrote a critical article listing problems he saw with the proposal. But roughly speaking, we can group them into two families of objections. And I think these are the two families that still hold today in the literature, where there's really a lot of debate going on. The first is Cannon says, well, the causal association between our bodily states and our emotions is one where there's a bit of a lack of clear smoking gun evidence. And his second objection is that maybe our bodily response is just not well suited to the complexity and sophistication of our emotions. You know, he's basically suggested our bodily responses are too specific, too insensitive, too slow to support the rich emotional lives of humans and animals. So if we go to the first objection, the causal question, first thing to bear in mind is that now that the theoretical framework has evolved to integrate the sense of the body with the broader context, of course, this in some part explains why there's a one-to-one relation between our bodily states and emotions, because of course we're integrating and inferring information from a broader set of contextual cues. But Cannon was right, and I think remains right to this day, that the actual causal evidence of what happens when our brain becomes partially unlinked from our body does indeed paint a bit of a complicated picture. So the most dramatic experiments done by Sherrington in the 1900s were done on dogs, where Sherrington removed the whole vagal and sympathetic system from the body of these dogs. Miraculously, they recovered from this, and even more miraculously, Sherrington saw very little change in their behavior. They still jumped around and were happy when they saw another dog they liked. They would still growl and become annoyed when a stranger came into the room. So this is obviously a big red exclamation mark saying, how is it possible if the body is so important in emotion for these dogs to apparently have normal, quote-unquote, emotional lives? If we now turn to the human literature, things are also a little bit ambiguous. There are cases in which intervening on the feedback from the body to the brain does have devastating effects. This is a report by a woman who underwent medical sympathectomy, so she no longer had sympathetic feedback from her body. I'll read a report because it's very tragic. What she says is, my emotions are blunted. There is an unsettling deadness and indifference towards my prior life, aspirations and goals. I would say that it has changed how I relate to people. I do not relate. So, I mean, here there seems to be very strong evidence that the emotional life of this woman has been deeply perturbed by the change in her sensations in the body. On the other hand, if we look at more systematic studies, for example, a lot have been done in spinal cord lesion patients, the evidence about changes in their emotional life is a little mixed. People tend to report some mild changes, but then backpedal a little. They don't seem very sure that there are really big changes happening. So this just to point out that the causal evidence of the link between the body and emotions has been hard to come by, and it seems to be quite ambiguous. There are very few black and white experiments that really show a causal link, even with very large interventions, as I've just showed you. What about the second objection? Second objection is that our body is pretty much an on or off, fight or flight, feed or breed system. And therefore, this cannot reflect the true richness of our emotions. This was really the view in Cannon's day, so back in the early 20th century, however, things have changed a lot. And I'll just point out two experiments underlying this, but I think they reflect a general trend. The first is a set of experiments in humans that have shown that there are quite subtle changes in the body corresponding to different emotions. Historically, one very important one done by Axe in the 50s showed that if you measure really lots of different bodily variables here on the x-axis, you can distinguish between anger and fear. And this is important because the view in Cannon's day was that the sympathetic system, when you are angry, so the fight system, or afraid, the flight system would just activate the whole sympathetic system, and there was no reason for there to be a difference. And after Axe's initial study, there was really a cascade of studies looking at many humans, saying that yes, there are sort of specific responses in the body to these specific emotions. And what about the sensitivity of our body? Here there's a little bit of a dearth of information about how the brain represents the body. We do know that anatomically, the information is getting there, and the information that gets there is quite subtle. So this is an example study I found very striking that analyzed the cells in our vagus nerves, our parasympathetic feedback to the brain. And remarkably, what they found is there are cells that are devoted to specific layers of specific organs, reporting specific variables back to the brain. So what's arriving in your brain is the pH and the pressure and the temperature of the inside and the outside of your liver and your heart and your kidneys. So it's very fine information getting there. But what we don't know as yet is how this information is used. Does it go up to the thalamus? Does it go up to the cortex? Does it impact emotional processing? So on the second objection, I think we've changed, the opinion has changed quite a lot, and we know that bodily responses are quite highly differentiated. The information seems to get to the brain, but it's not clear what the neural mechanisms are exploiting this. And what I wanted to highlight in the introduction is I think this lack of causal evidence and the lack of neural mechanisms are things that can be well addressed in the animal, particularly the mouse, which is the animal I work with, because we do have quite a lot of tools giving us handles on these aspects. And so what I wanted to talk to you about today is this, going back to a much simpler scheme of what's going on, some investigations we've been doing in how somatic changes in the mouse can influence emotional behavior. And there are going to be three parts to the talk. First, I'll present you some work we've been doing trying to understand if there is indeed a causal link between certain somatic states and emotional behavior. Second, some work addressing how specific bodily states are in the mouse and how if they correspond to specific emotions. And finally, a little bit preliminary data on how the brain mechanisms could actually read out this somatic information to implement behavior. So with that, I'm going to jump right in, but I forgot to say, if you have any questions and want to interrupt, please do before I get to the actual data. No, we want to stay on board, hopefully. So how do you study emotions in a mouse? So, of course, what we need is a reliable way of evoking emotional states in a mouse. One way to do this is to frighten the mouse. They're going to display quite stereotypical behavior. So in this study, we used really the bread and butter experimental paradigm for fear research in mice, which is tone fear conditioning. So you take two tones, what I'm going to call the CS minus and plus. You habituate the mouse to these tones, and then you take one of the sounds and you associate it with a mild foot shock. And surprisingly, the next day, if you present this sound, the mouse is going to change its behavior because it's learned to associate the sound with the disagreeable foot shock. And the most predominantly studied behavior is freezing. And if you present the sound and the mouse becomes completely immobile, which is a behavior that makes a lot of etiological sense, because if you're a prey, if you stop moving, you have a lot less chance to be detected by your predator. What we observed is during this freezing behavior, the breathing of the mouse underwent a radical change. So here is breathing of a mouse navigating its environment. It's breathing about 10 hertz. You can see it's quite an irregular breathing. And then the mouse begins to freeze, and we see this very regular 4 hertz breathing develop here in all the mice that we studied. And when we saw this, we were quite excited because we knew pathways in the mouth by which the breathing oscillation could feed back to the brain. So how does this work? If you look at some data here of a mouse breathing in and out, so in gray you have respiration. As it breathes in and out, of course, air goes in and out of its nostril, changing the pressure in its nasal pathways. And in its olfactory bulb, there are mechanism receptors that are going to sense these changes in pressure and therefore translate the breathing oscillation into an oscillation in its olfactory system. And therefore, as the mouse begins to freeze here, you see this beautiful 4 hertz respiration, which translates into a really regular oscillation in its olfactory system that you can see here if we look at the Fourier transform with a very strong peak around 4 hertz. So we go from the respiratory system to the olfactory system. And what we knew from the literature is that this oscillation could then be transmitted to pretty much the whole of the mouth and brain. The cortex, the hippocampus, the thalamus, people have documented this over the past 20 or 30 years now. And one of the areas where the respiratory rhythm was known to occur caught our attention is the prefrontal cortex. The prefrontal cortex in mouse, and also in other animals, is known to be heavily involved in regulating emotions. Particularly in the mouse, there are a lot of studies showing that the prefrontal cortex can regulate freezing. And we thought to ourselves, maybe this 4 Hertz oscillation can impact freezing via the entrainment of the prefrontal cortex. First, we had to check that our oscillation was indeed there. So here's another way of looking at the oscillation in the olfactory bulb. You can see time here along the x-axis, and along the y-axis there are frequencies, and the fact that you see a very red band here during freezing indicates that the mouse was breathing at 4 Hertz, and we see this in the olfactory bulb. If we look now into the prefrontal cortex, we also see this very strong oscillation, and if we measure the coherence, that is whether the two are oscillating together, we see a very strong coherence. And to test that the olfactory bulb is really the region relaying the oscillation, we remove the olfactory bulb by lobectomy. And if now during freezing you look at the prefrontal cortex, the oscillation has disappeared. So what this shows is that prefrontal cortex local field potentials are indeed entrained by the respiratory rhythm. But we wanted to know whether this was changing how the neurons were behaving themselves. So to do this, we recorded single unit spiking in the prefrontal cortex, and we asked whether they were changing their firing patterns based on the 4 Hertz oscillation. The one way to look at this is to look at the phase of your oscillation, so here from 0 to 2 pi, and then the same phase repeated up to 4 pi, and simply count when the spikes occur during the phase of your oscillation. And what you see in this example unit in the prefrontal cortex is that it has a very strong preference to fire just after the peak of the oscillation, just after pi here. And this is stronger in the freezing state than in the active state. Overall, we found that around 45% of units in the prefrontal cortex are significantly modulated by the 4 Hertz oscillation, and it always tends to be stronger when the mouse is freezing than when it's active. Again, to test the role of the olfactory bulb, we removed the olfactory bulb, and we found that the number of significant units dropped, and the very few units that were still entrained had this very messy entrainment here, as you see in the best unit compared to the best control units. So altogether, what these observations show is that when a mouse is afraid, it freezes. During freezing, its breathing changes to 4 Hertz, and this respiratory rhythm is going to entrain the olfactory bulb, which in turn entrains prefrontal cortex activity and is going to temporarily organize firing in this region. Which of course then raises the question, does this have any importance for behavior? So to do this, we could manipulate the olfactory bulb as the relay of this respiratory rhythm and observe if this changed the behavior of the mouse. So first we use the bulbectomy that I've already mentioned. This is to give you an idea of what it looks like. This is a mouse brain in a sham mouse with very large olfactory bulbs at the front of the brain. And this is a brain of a mouse after bulbectomy. And what we found is that overall there was a drop in the freezing levels of mice after removing its olfactory bulb. However, of course, this is a massive intervention. Mice rely massively on olfaction. It's a very big surgery, as you can tell by the photos. And so we wanted to complement this with some more precise manipulations. So to do this, we did optogenetics in the olfactory bulb. So we express a protein in the olfactory bulb that allows us to control the neurons in the olfactory bulb cells. And basically by flashing the light on and off, as you can see here, we can come and we can scramble the oscillation that is endogenous to the olfactory bulb. So you can think of this as normally the olfactory bulb would be nicely transmitting the 4 Hertz, and then when we're coming on top and adding these extra oscillations and so scrambling the information. What we saw when we did this in our control mice was no effect at all, but in the channel rhodopsin mice, we again see a drop in freezing, as you can see here. So this does seem to confirm that the breathing rhythm relayed to the prefrontal cortex impacts the behavior of the mouse and therefore has a causal role. However, if we really think about what's going on, this may not make a lot of sense, because on the one hand, I've told you, well, the mouse has to freeze to have the 4 Hertz oscillation, but you also need the 4 Hertz oscillation to get freezing. So this suggested to us that, maybe we hadn't fully understood how the system works. So we went back to our data and tried to understand what exactly was going on during the mouse's behavior. So there's a very simple way of looking at the mouse's behavior during this task. It's to construct a Markov model with two states. The mouse is either freezing or it's active, and with certain probabilities it's going from one to the other. So for example, here the mouse is active and it has to choose, do I start freezing or do I stay active? And if it started freezing, on the next step it has to choose whether to maintain freezing or switch to an active state. So what this means is that we have a very simple model with two parameters that can be easily calculated that regulate if the mouse is going to initiate freezing and then if the mouse is going to maintain freezing after initiation. So what happened when we manipulated the olfactory bulb? What we find for both our manipulations is that initiation is not impacted at all. So my start freezing in exactly the same way with or without the respiratory feedback. But what does change is their ability to maintain this behavior. So once they've begun freezing, it's then that 4 Hertz oscillation becomes important and regulates whether they stay in the state, whether they become active again. So I think and hope you would agree that this paints a picture that makes a lot more sense. The mouse begins to freeze, it modifies its respiratory rate. This respiratory rate feeds back to the brain, recruits activity in the prefrontal cortex, and then can recruit the prefrontal cortex regulatory ability to decide whether to maintain or initiate freezing. I think this fits also a lot better with the way we think about top-down regulation from cortical regions onto emotional centers, where it's not the cortex that's going to initiate these emotional states, but the cortex can then regulate and modify them in a more context-dependent manner. So interestingly here, we have quite a clear causal case for a role of bodily feedback in regulating emotional states, but it only impacts a very specific aspect of the emotional state, namely in this case, the maintenance. And so going back to what I underlined earlier in the talk, that is the rather ambiguous causal evidence about the relation between the brain, the body, and emotional states, it may suggest that what we should look for, either in animals or in humans afflicted with a lack of bodily feedback, are specific aspects of emotions that are going to be dysregulated instead of a lack or a general dampening of emotions. So from our results, for example, one prediction we would make is that people lacking bodily feedback should have different temporal structure of their emotions. For example, shorter-lived emotions. And actually, if you go back to the study by Homan in the 60s, and you go through what people say, with only a little bit of cherry picking, I promise, you can find quite a few quotes that do suggest that these people have emotions, but that maybe they get over them a little bit quicker. And I've underlined some of these quotes here. But without pushing this too much, I think it does make an interesting prediction that perhaps could be tested in humans. So to wrap up this first part, what I've illustrated are some causal links between breathing and fearful behavior in the mouse via feedback of this oscillation via the olfactory system. And I'd also like to take a moment here to point out that since this is feeding back via the olfactory system, all the classical lesions that remove sympathetic systems or vagal systems are not going to impact this feedback pathway. And that, I think, is another thing we need to think about when we're thinking about the causal role of the body, is that it has many, many pathways to impact the brain, even beyond the sort of classical sympathetic, parasympathetic systems. And so to really get a causal hand, we have to have a very clear picture of what pathways exist and how to manipulate them. So I focus on breathing, but I would like to point out that there are some other interesting studies in the mouse done by other laboratories looking at the vagus nerve and the heart. And they too found that it really was specific aspects of behavior that were impacted. In the case of the vagus nerve, it seemed to be the extinction of freezing, but not the other aspects. And in the case of heartbeats, very recent studies showed that this only had an influence when the mouse was already in an anxious state, but not in a baseline state. So with that, I'd like to get on to the second part of the talk, because what I've shown you so far, if you have a skeptical mind, might not really have convinced you that I've shown you that breathing is related to anything emotional. Because another big difference between an active mouse and a freezing mouse is that one mouse is running around and one mouse is immobile. So what I may have shown you is just that breathing changes when locomotion changes, which would not exactly be a scoop. So in the second part, I want to illustrate some work we've been doing, trying to sort of really get at whether or not these bodily changes are linked to emotional changes. So to do this, we changed our paradigm to a paradigm that could evoke a broader and more complex range of emotional states in the animal. So this paradigm is something called the U maze. It's a very simple task. You have a maze shaped like this with two arms that we're going to call the shock and safe arm. Initially, nothing happens. The mouse just runs around very happily, explores both arms. And then after a little while, once it enters the shock arms, after a few seconds, it's going to receive a mild shock. And what we observe is that the mouse tends to avoid the shock zone and spend more time in the safe zone. But interestingly, what we found is that the mouse's behavior becomes highly organized throughout this maze. So here I've linearized the maze, where 0 is the shock zone, and 1 all the way on the other side is the safe region. And you can see that if we look at different behaviors, such as jumping up and down, or risk assessment, or grooming, the mouse displays these behaviors in a very organized way throughout the maze. And when we saw this, it immediately made us think of some historical studies looking at the ethology of fear. So if you look at how animals respond to predators, ethologists have defined a predatory imminent scale, going from the highest level here, where the cat is just about to eat you, all the way over here to a situation where maybe you can just smell a cat, or you thought there was a cat. And what they found is rather systematically, mainly using studies in rats, but it's been confirmed in other animals, that along this predatory imminent scale, you get very stereotyped behaviors. And even more interestingly, by using in particular pharmacological manipulations, they linked this predatory imminent scale to panic and anxiety as it has been described in humans. So in humans, panic disorder and generalized anxiety disorder are treated by different classes of drugs. And so what the ethologists did is to evaluate whether these panicolytic and anxiolytic drugs, as defined in humans, affected the behavior of the animals in different ways. And they indeed found that panicolytics mainly had an effect over here, whereas anxiolytics mainly had an effect at the lower scale of predatory imminence. So after seeing this in the maze, we thought, well, maybe the maze is sort of a nice way in the lab of getting at this predatory imminence scale. However, we had one problem, and the problem was freezing. So instead of this highly organized structure that we got with other behaviors, we found freezing all over the place. Basically, in every single corner of the maze, at about the same level, the mouse displays freezing. And really, it is exactly the same behavior of complete immobility as measured here with a very sensitive head accelerometer. So this raises the question, is freezing, contrary to other behaviors, completely homogeneous? However, what I've shown you so far, we looked at the global behavior of the mouse. What if we look at what's going on inside the body? So here is some example data, time again, and on the x-axis, frequency here. And we're looking at the olfactory bulb spectrogram, but you can think of this as a proxy for the way the mouse is breathing. So first, the mouse is running around the maze exploring. It enters the shock zone, gets a shock, and freezes. And you can see here the oscillation becoming quite strong at around 6 Hertz. And I've illustrated this here with an example data. The mouse then gets a second shock, decides it's time to leave, runs away to the safe zone, and again begins to freeze. And I hope you can appreciate on this example that the frequency here is about 2 Hertz below the frequency we saw in the shock zone. And the difference is quite clear in the raw data. And this is something we saw for all of our mice. They have different breathing rates depending on where they're freezing. So a first indication that no, this freezing behavior is not homogeneous throughout the maze. So I won't belabor the point since we're focusing on breathing today, but we get similar results if we look at other bodily variables. So we looked at heart rate, which is also very different between the two regions. And we also looked at blood flow by monitoring it indirectly by a temperature. And we also see that there is a difference between these two types of freezing on the shock and on the safe side. So if we put this together, although we have identical behavior, now we really have different bodily states that putatively correspond to quite different emotional states for the animal. Since in one case, it's at a very high level of threat, and in the other, it's instead in a safer region of the environment. And going back to the ethological study I introduced, we wondered whether these two states of freezing might not be proxies for something panic-like and anxiety-like in the animals. And so to test this idea, we decided to use a panicogenic and an anxiogenic and measure what kinds of behavior they may appear, and then compare them to what we had in the humans. So first we used a panicogenic. The panicogenic we used was stimulation of the dorsal periaqueductal gray. So this is a region that's known to be strongly associated with panic. For example, in humans, if a person breathes in CO2, they have the impression of suffocating, which of course provokes a very strong panic reaction, and you'll see the PAG light up in imaging studies. So we stimulated this region in the mouse. The mouse began to freeze, and what we observed is the freezing tends to occur at around 5 or 6 hertz. So exactly the frequency I showed you, where the mouse is breathing at around 6 hertz, it looks very much like the shock zone freezing. What about an anxiogenic? So in mice, it's known that a specific concentration of nicotine injected to the mouse is going to elicit anxiety-like behaviors as measured by classical behavioral tasks like the plus-made or the open field. So we injected nicotine to these completely naive mice. They also showed some freezing, but in this case, the breathing was quite different from the DPAC stimulation. Instead, we had breathing at around 2 to 3 hertz. So what we see here are two different treatments, panicogenic and anxiogenic, that elicit body states that are very reminiscent of what we saw in the humans. So we took this to be evidence that in a completely identical behavioral state, so we get rid of the problem I mentioned of locomotor biases in interpreting our data, we can see a mouse freezing in two different body states, and we do have a little bit of evidence to suggest that these are maybe panic-like and anxiety-like states. So suggesting that, yes, in the mouse, in this particular paradigm at least, we can identify two emotional states defined by different bodily states. And in the last part of the talk, I'm going to try and harness this because I think it really gives us a really interesting opportunity, since we've gotten rid of all the gross behavioral biases, of studying how the brain might interpret these different bodily signals and perhaps tie them to different emotional states. The data I'm going to show you is a little bit more speculative and preliminary, and I would really love to have feedback on whether you think these ideas hold up. So the question I'd like to ask is, since we've observed these two different body states, is it possible for the brain to monitor the emotional state of the animal simply by monitoring its bodily states? So we're going to focus on the prefrontal cortex because A, we had data lying around, and B, I've already shown you that it seems to have some causal involvement. And basically, what might be happening is this. Imagine you have neurons in the prefrontal cortex that don't know anything about the emotional state of the animal, that don't know anything about the humase, but they are tuned to different breathing frequencies. You have these neurons that respond to 4 hertz, and these neurons that respond to 2 hertz. Then for free, these neurons are also going to fire either in the panic-like state or in the anxiety-like state. And so you could have an interoceptive readout providing information about the emotional state of the animal. So to test this idea, I'm going to show you some analysis that addresses 3 questions, one after the other. First, for what I've suggested to be true, the PFC has to differentiate between the shock side and the safe side freezing, has to encode the emotional state. Second, it has to encode breathing rates. And if these two are true, we can then ask whether the encoding of the breathing rate can drive the coding of the emotional states. So let's start with this first question. Do we see in the prefrontal cortex firing reflection of the panic-like and anxiety-like states? So here again is an olfactory bulb spectrogram, a section of our data from the UMAs. Here the mouse is freezing on the shock side, getting a lot of shocks, but staying on the spot, decides to leave, and goes and freezes on the safe side at this lower breathing frequency. We look at the firing of prefrontal cortex neurons, we see This neuron doesn't really care where the mouse is freezing, it's just inhibited as soon as it freezes. But this unit and this unit seem to have a preference for freezing side. This unit fires when the mouse is freezing on the safe side, whereas this neuron fires when it's freezing on the shock side. And then we can take the full population of neurons and ask how accurately can we guess, based on their activity, where the mouse is freezing. And we see that with about 80% accuracy, depending on the number of neurons we recorded, we can indeed decode where the animal is freezing. And importantly, this decoding is better during freezing than during movement. And we have another analysis to suggest that it's not just that the prefrontal cortex is encoding the position of the animal in the maze, really something specific to freezing. So taken at naively, this analysis could suggest that yes, the PFC does have information about which kind of emotional state the mouse is in. Now, do we see encoding of breathing rates in the prefrontal cortex? So here, of course, it's absolutely crucial that we do our analysis outside of freezing, because otherwise the different kind of freezing and the different kind of breathing are so tightly correlated we won't be able to conclude. So what I did is I took a period while the mouse was just navigating in a neutral context, no shocks, no stress, and looked at how the neurons in the prefrontal cortex fired at different breathing frequencies. And this is what we observe for the neurons that seem to be significantly tuned, is that we get neurons that respond to low breathing frequencies, neurons overwhelmingly that prefer the high frequencies, and some neurons here that respond to the medium frequencies. And I should just point out that this is being cross-validated on different part data. So the fact that the neurons do have a peak is not just because I aligned the data. So overall, this concerns about 45% of neurons. Just to show you some example neurons here, we do get in some cases quite nice tuning curves that, you know, naively might be reminiscent of what we would see in the auditory system if we were looking at sound frequency and not breathing frequency. So there does seem to be information in the prefrontal cortex about the current breathing frequency. However, once again, we are plagued by correlations. Breathing is going to be correlated with what the mouse is doing, how fast it's going. And so what could seem to be a breathing tuning curve might just be a speed tuning curve or a direction tuning curve. So can we assess how well this tuning generalizes between behaviors? So to do this, we did the following analysis. This is just a schematic, so that's why you see the two same pictures here. So we take the tuning of the neurons in one state while the mouse is running around and exploring. And then we take it in a completely different state when the mouse is immobile. So I did the analysis either for freezing or for the sleep state. And what we can ask is whether the representation, let's say at 2 Hertz during exploration, and the representation at 2 Hertz during sleep are similar or not. If they're completely different, it suggests that this representation is not reliable and it's maybe linked to some other variables. But if they're very correlated and very similar, despite the fact that the mouse is doing completely different things, it would suggest that there is a reliable tuning to breathing in the prefrontal cortex. So we can do this correlation for every single breathing frequency, which gives us a correlation matrix like this. And the yellow color means that they're strongly correlated. And what you can see is that overall, we do have, for similar frequencies, similar representations. So to give you an idea, if the representation was perfectly preserved, you would have one yellow line along the diagonal. And if it was random, there would be no difference in color at all. So there is some preservation, even though here the mouse is exploring and here it's freezing. And if we do the analysis both for freezing and for sleep, in both cases, we are significantly different from if the PFC was just randomly encoding breathing in these states. So hope this convinces you that we do see some encoding of breathing rate in prefrontal cortex. Maybe it's only a subpopulation of these neurons that are actually encoding it reliably between states. But at least in this subpopulation, there is information about breathing rates in the prefrontal cortex. So now that we've checked our first 2 questions, we can ask whether this interoceptive encoding may in part explain the emotional encoding in the prefrontal cortex. So to do this, we go back to our representation of breathing in the neutral context. And from this, we can extract a single axis in our high dimensional data that maximizes the difference between high frequency and low frequency breathing in this neutral state. Now, if the PFC is exploiting this axis to decode shock versus safe side, then we expect that the neurons here that are tuned to the low frequency of breathing in a neutral context will be the neurons that light up when the animal is on the safe side. And contrary-wise, these neurons that prefer the high frequency are going to light up on the shock side. So what we can do is we can take the axis from this neutral context, project onto it data from the freezing context, and ask whether this immediately tells us where the mouse is on the shock or the safe side. And when we do this, we find that with about 75% accuracy, we can indeed tell whether the mouse is on the shock or the safe side, entirely based on the PFC tuning to breathing in a completely different context. And just remind you, this is only about 5 or 6% lower than what I showed you earlier when we were really optimizing the decoder to tell the difference between the two. So this is all correlative for the moment, but I hope it suggests to you at least the possibility that tuning to interoceptive variables may be a mechanism by which the prefrontal cortex can track the emotional state of the animal in this context. And so that wraps up the third part. Overall, what I wanted to illustrate to you today was in this very specific context of fear in the mouse, that we do seem to be able to pin down some definite causal links between body states and emotional regulation, that these body states can be quite specific to different emotional states. And I hope I suggested to you a mechanism by which the brain could exploit the specificity of these bodily responses in order to read out emotional states. And moving forward, I think it's this last part that holds a lot of promise for trying to sort of understand how the body impacts emotional states and maybe bring us back to this more complicated picture that I presented in the beginning. Because I think the crucial question is how data about the body is integrated with context and beliefs and the global situation. But to understand how it's integrated, we really first need to understand how this interoceptive readout works. How precise is it? How reliable? Where is it in the brain? Is it context dependent or does it depend on context? Because the answer to these questions are going to condition to what extent the brain can identify emotions from the body, the information it can extract, how it can relate that information to other aspects, and in fine, how it can use that information to regulate behavior. So with that, I'll be quiet. But first, of course, I wanted to acknowledge all the people that participated in this work. First and foremost, Karen Benjinan, who supervised and co-worked with me on everything, and in particular, Julie Lefort, who is co-author on the first study, and Baptiste Maillot on the second one, who both had a lot of input for all of this, as well as all the current and past lab members for their discussions and advice. And thank you for listening. I'd be delighted to take any questions.